it's a pleasure to be here. Thank you very much for the introduction. My lab for a long time has been into seasonal timing. And, plants need to get what they do right in the seasons because they can't get up and move around. So they spend a lot of time figuring out what season they're in and changing what they do. And of course, seasons, depending where you are in the world, are then different. So for now, almost 30 years, we've been trying to get to mechanism and natural variation understanding, seasonal timing, mechanism, and then how that's changed as plants have adapted. Now, the system we use right from the beginning was Arabidopsis thaliana, a system where we could do the molecular genetics. But Arabidopsis, there are many types of Arabidopsis. Sort of most people see Arabidopsis as this rapid cycling type. That's sort of what everybody knows it as. We can do our rapid genetics. But in fact, Arabidopsis has these winter annual types, and I'll show you there's a whole range in between. And so Arabidopsis is a nice example of basically having a choice of life history. Some of them overwinter before they flower, and others have this rapid cycling type, and there's a lot of biology as to which habit is most appropriate for which climate and which is fitter, etc. But clearly, the plants have to get timing right, and this is the timing to reproductive development, which is we study flowering. Because if they get it wrong, reproductive success is disastrous. So very strong selective pressure. So, as I say, right at the beginning, I decided, yes, that's what we wanted to look at, was how do plants overwinter? How do they, some need to overwinter and some don't? If they need to overwinter, how do they actually know they've experienced winter? What is winter to a plant? And how is that varying in different places that Arabidopsis grows? So we did lots of analysis, and this is showing some of the natural variation in this winter requirement, which is a process called vernalization. Vernalization is the actual acceleration of flowering by prolonged cold. Very important for most of our crop plants. You've probably heard of winter and spring-sown wheat and brassicas and oilseed rapes. They differ in their vernalization requirements. So there are winter-sown types need to overwinter before they will flower. They require winter to flower. And so if you take Arabidopsis successions, and there was lots collected from lots of different places around the world, and grow them in a warm cabinet, you see a lot of variation for flowering time. So this is just put in the cabinet, looked at when they flower. You see lots of variation. There's the rapid cycling types going all the way up to these really very late types. This is 120 days to flowering, and they still haven't flowered. So basically, they absolutely need vernalization. Whereas if you look at the response to the cold, this is now with an 8-week, you can see within those obligate requirers, you have a big variation. So at the beginning, we had a lot of natural variation then in whether they needed to overwinter and their ability to respond. Now, at the time, and a lot of this early work was a collaboration with Magnus Nordborg, who has really developed the natural variation work in the Arabidopsis thaliana field, has sort of set up doing all the GWAS, and he thought this would be a kind of a perfect opportunity to look at the molecular basis of adaptation, because there was a lot of natural variation in it. Plus, we had a lot of information about what control flowering time in Arabidopsis. So at the beginning, and this was one of the first things my lab did with lots of interactions from lots of other people, was to sort of take Arabidopsis mutants now, EMS-induced generally mutants, and define the genes that require control flowering time. Now there's lots of pathways. There's clocks involved, there's light quality, there's photoperiod. These plants are monitoring lots of different aspects to judge when to flower. One interesting thing is that all of these different pathways, and in fact, if you're a gardener, some people think, okay, well, this plant needs light, and this plant species needs temperature, and this plant species in the desert needs water. So at the beginning, everyone thought that flowering was going to be regulated rather differently in response to these different physiological cues. But this was one of the first realizations that all of these inputs, these different environmental inputs, converged onto a common set of targets. And it was their quantitative activation that then precipitated the transition to flowering. So that was an interesting first finding. Because we had lots of genes here that potentially were going to be targets for this natural variation. Now, this is a simple version, quite an old one. This is actually what it looks like. And I just want to make the point, there are lots of genes that when you mutate will affect the flowering time, okay? You don't have to read any of it. There's been lots of discussion about, is Arabidopsis network relevant for any other plant species? Because, of course, they seem to flower in response to different cues. Well, yes, indeed, actually, and this is the valiant attempt of an ex-colleague of mine, Dave Laurie, to try and line up rice and temperate grass flowering control. And in fact, you don't really, some of these have even got different names just to confuse everyone. But basically, the networks look rather similar. The wiring is really what's changed. So anyway, just to say, lots of genes. So then the question was, can we use these genome-wide association analyses type activities to actually then look at the molecular basis of adaptation, of flowering time variation? And this was, to say, this long-term collaboration with Magnus. We used 2 populations. He had collected something which everyone calls a set 1, set 2. There was 384 lines of these, and he'd selected them very carefully to represent local diversity as well as worldwide diversity to try and set this up so that he could do the association analysis. And then following that, we actually also went in and found a more localized population just from Sweden. So all these little green dots here are where the Swedish set were localized from. So, this was back in sort of early 90s, so we were very optimistic, thinking this is going to be great, we're going to have lots of GWAS hits for flowering time. And in fact, it was really not terribly successful for flowering time variation. And the worldwide set, there were lots of phenotypes mapped. This is one of the papers. But for flowering time, in the end, the outcome was that because we have the corrections of population structure, we sort of lost associations for flowering time. Hence, then we went to this localized Swedish set. So actually, we then did it all over again, both flowering time, vernalization, all sorts of things. And in fact, again, that wasn't terribly successful. And by the end, I hope you'll realize why. But the answer is actually because of massive allelic heterogeneity at a few loci, where basically you get one gene has been used over and over again with different causative mutations. So you lose all the power to detect that SNP with a phenotype, because the phenotype is associated with lots of different SNPs at that same locus. And this is a really fantastic paper that Magnus published a couple of years ago, which basically says that. And the plant community has now sort of struggled through this, and we've done the QTL analysis, because of course in plants you can do the crosses with exactly what you want to cross with what. So we actually got the answer. So I just put this on to reference this and perhaps refer you to it. Anyway, so at the same time as doing this genome-wide association analysis, we were just doing the standard crosses, QTL mapping, to actually ask, so okay, if we take these different types, what happens? What do we find in terms of the determinants? So we picked, I just show a subset here of all of these hundreds of ones that we looked at. We thought, okay, let's take the ones with the weirdest looking vernalization response and then map what's underlying that. So we just took them. This one, for example, this orange one, you can give it 4 weeks cold, this particular accession, have absolutely no effect on flowering at all. Then it starts to accelerate. This is, leaf number is our proxy for measuring, so the fewer the leaves, the faster it flowers. You see that there are others where basically this green one, it is really very slow, very sluggish indeed. So you can almost give it 10 weeks cold and it's only just about getting to the point of a small number of leaves. So basically quite distinct vernalization responses. But when we did the QTLs, all of them basically showed that the major QTL mapped to this one gene. And this one gene was a gene called flowering locus C, FLC, which is probably the topic most of the rest of the talk. And it's the simple way of thinking about it. It's a brake to flowering. If the plant is expressing FLC, it stays vegetative. It's a Madsbach transcriptional repressor that keeps off all of the genes in the meristem that's required to switch to a floral fate. So in fact, most of what I'll talk about is how that gene is switched off by the cold, this epigenetic silencing of FLC. But actually, the winter rapid cycling habit is also determined by a quantitative transcription of FLC, and right at the end, I'll just get back to that. So, in fact, all these genes could have been used, but somehow, actually, this is the major determinant that we found. Now, when we then went and looked at the sequence of FLC in a number of these accessions, actually they fell into kind of quite nice little groups, and that was whether or not we sequenced from the whole worldwide population or, in fact, from this local Swedish one. We see these very distinct groups where within a group there's really not much polymorphism, but you see quite a lot of divergence between the groups. And all of the SNPs are non-coding. So the amino acid sequence of all of these FLCs from all of these accessions is identical. So that kind of suggested that that the protein was clearly quite conserved and very strongly selected for. And then when we looked at all of our, do you remember the graphs I showed you with these very distinct vernalization responses, when we looked at where they were on our tree, we'd managed to pick one accession from every one of these different groups, suggesting that these non-coding SNPs were actually affecting the regulation of FLC. Now, to really prove that, so I've color-coded them, and SV is slow vernalizer, rapid vernalizer. So we've got different rapid vernalizing types and different slow vernalizing types shown here. We then cloned each one out of the different alleles, put them by transgenesis into a common background, and then looked at a sort of vernalization assay, no matter how we did it. But this is now those different alleles in a common background versus the vernalization expression in that natural accession, with a very remarkable correlation. So in fact, most of the vernalization response variation maps to those non-coding SNPs. So an obvious question was, well, is there any sort of obviously geographical stratification to where those different haplotypes occur? In fact, this is now this color coding, RV, rapid vernalizing, S, slow vernalizing. This is their distribution. When we look at the centroid of each of those, this is what we see up here. There's one which is often up in the northern latitudes, this SV4 type, but there wasn't an obvious latitudinal decline or anything like that. So it was obviously much more subtle than that for whatever was causing or potentially functionally associating with the variation of these in the natural populations. But when we looked at, and this is Magnus's analysis with now a 250K SNP chip to look at the polymorphism around FLC, so this is now aligned so that this is the FLC region here, and this is a 50 kb either side of FLC lined up with the SNPs to sort of put FLC in the group. So here's the FLC locus. You can see that, in fact, the different haplotypes, as I've shown you before, have these long haplotype blocks, which is relatively unusual in the Arabidopsis genome. These blocks are usually a lot shorter than this. So that was suggesting there was some maintenance of those blocks. There was reduced recombination in that region for some reason. But it did explain quite nicely why we really weren't detecting it in GWAS, because, of course, we had SV as a phenotype contributed by very different molecular SNPs. Now, the next question, of course, is, well, what do those SNPs, what's that natural variation doing to the mechanism? So I'm just going to go into a bit of mechanistic detail, and then I'll come back to explaining how those SNPs affect the mechanism. So this is vernalization, sort of classic picture, in that the plants, when they don't have any cold, are expressing FLC at a very high level. Plants are vegetative. But then with increasing exposure to long cold, weeks of cold, you see this slow, progressive shutdown of FLC expression. And it's then epigenetically maintained at that low level, even when the plants come back out into the warm. And that, of course, allows now, so you've removed the break, and so the plants can now respond to the long days and the ambient temperatures of spring, and flowering can be induced. But it's interestingly in that if you take a plant that's had 2 weeks, it will then stay at that level that's 2 weeks versus the level that's 4 weeks. So the quantitativeness had an epigenetic memory. Now, we went in and tried to define what the basis of that was many, many years ago now by creating mutants which could no longer vernalize properly. So we took a wild type which required vernalization, did mutagenesis, and vernalized most of the M2s, and then the ones that were late were these, something was defective then in the vernalization response. And this took us into the world of chromatin silencing, polycomb repressive complex 2, which now is a well-known chromatin silencing mechanism in most higher eukaryotes. Sort of involved in affecting histone modifications, the lysine 27 particularly, and it's always associated with silencing of genes. So that was quite nice in terms of a memory mechanism. We also had 2 PhD proteins which were required for vernalization. These are plant homeodomain. These PhDs actually are found in all organisms, but they just happen to be first characterized in plants, so they're hence PhD their name. They tend to bind histone modifications. And then there was a homologue of an HP1 protein, again, another well-characterized chromatin protein called like heterochromatin protein 1. So all of these were suggesting a chromatin silencing mechanism. But we went in and said, well, why is it quantitative? What's the quantitative basis? So looked, knowing that it was a PRC2 phenotype, at K27 trimethylation over now the body of the gene, and here's our FLC gene. These are the exonic regions. This is about an 8 kb region. And what you're looking at here is the chip with looking at K27 trimethylation within plants that had no cold, which is black, or with increasing exposure to cold up to 8 weeks. So this very nicely gave a quantitative accumulation of the chromatin silencing mark, which at one point said, oh, that's great, we can explain the quantitative memory. But actually, when you think about it, how on earth can a plant, when it's had partial vernalization, let's say 4 weeks, how can it inherit that level of chromatin silencing in a quantitative manner through many cell divisions? And in fact, through modeling, and this was actually when we started to work with my colleague now, computational biologist Martin Howard at John Innes, he was saying, he said, he's a theoretical physicist, so he thought about this very differently. He said, I think you'll have got a switch, a binary switch. It'll either be silenced or active, and it's just a proportion of cells that you're looking at that's changing. And that was exactly right. That's what explains the quantitativeness of vernalization. So if you look at a plant, and I'm actually going to show you a picture of a root, which is why there's a schematic of a root here. So here's a root with no cold. We have a traffic light system, so gene on, green, FLC is on. And slowly, as the plants are exposed to increasing weeks of cold, you have a switch in a gene, at the locus in a cell, and I've drawn one here actually in the meristem. In the cold, the switch is happening. You then bring the plants into the warm again, and the roots grow. And then the root is a nice system in that you have only anticlinal divisions in the root. And so this is a developmental lineage of cells from a single meristem cell down here. And what we could see, and I'll show you in a minute, I'll just explain it schematically, is that the cells that had switched then stay off, and they can be next door to a cell which hasn't switched, which stays on. So it's a cell autonomous switching mechanism that's happening with increasing probability as plants are exposed to increasing cold. So this is the real data. We've got a venous fusion, so we can mark FLC. It's a nuclear protein, so you can see it going into the nucleus. Here's a root where you've got everything expressed. But as the plants now are getting more and more cold, as you grow them out afterwards, there's 7 days at the end of the cold, so the root's grown. Now you can see here's this one where basically this hasn't switched off, and so all its daughters, granddaughters, etc. stay on, whereas the file next door is off. Now, I always forget to say this, but FLC regulates, the downstream target of FLC is a molecule called FT, which moves around the plant. So in fact, this digitalness of exposure to cold is then averaged out by the target, immediate target downstream, which then moves around the plant. Okay, so we had a digital switch, or a digital memory, I should say. What about the actual switching mechanism? Now, I'm going to whiz through this quite quickly, schematics, because I want to get back to the natural variation. But the picture we have then is that we have FLC expressed in autumn. The gene is on. You have a silencing quite quickly, transcriptionally in the cold. So you see a decrease in what we see as the opposing chromatin state to polycomb, which is actually K36 trimethylation. So this goes down as the polycomb goes up. And so this is one cold step. But the second cold step is this where these PhD proteins come in. This is one of them, it's Vin3, which is also cold-induced. But it takes a long time to induce Vin3 to its maximal level. And that drives nucleation of this modified polycomb complex. So here's the PRC2, this orange thing here. And there's 2 PhD proteins and actually a DNA binding protein called Val1 that localizes just at one site in the intron of the gene. And that's what happens in the cold. That binds, and that follows. You need to transcriptionally silence the gene first, and then you get nucleation of this chromatin polycomb silencing. It accumulates in the cold, and that's the switch in the cold. That confers a metastable switch. You then, as the plants come back into the warm, this polycomb spreads across the body of the gene. And I showed you the K27 spread across the whole body. And you need that spreading to actually maintain the silencing for the long term over the many cell divisions that happen in the spring and the summer. So it's interesting, what's this switch? Why is it here? Well, when we went in, again, the mutagenesis has been so useful for this. We had a mutant, which was a single SNP in the intron of FLC here, which was completely blind to vernalization. And so using that mutation, we actually then went fishing with what bound to it. It was 2 motifs nearby to each other. And actually that binds a DNA binding protein called Val1. None of these are specific to flowering at all, I should say. Actually, FLC is remarkable in that everything we work with in a trans-factor way is regulating many thousands of genes in the Arabidopsis genome. It's just FLC is a very sensitive target. So this DNA binding protein then brings in modifiers that actually modify co-transcriptionally RNA processing, and this brings in, and we sort of went fishing with this pulls out this complex, and these pull out now. We get back to the PhD proteins and the polycomb PRC2. So we've tried to link the genetic data with the molecular who touches who to work out a sequence of events in this nucleation. Now, what's interesting, though, is this DNA binding protein, you kind of imagine, okay, well, you're familiar with this. Here's a trans factor binding to a cis element. But this switch occurs in a cis-dependent way. And when we look, and I refer you to this paper if you're interested, with different markers fused to FLC, we can look at multiple copies in the same cell. We can show that this is a cis-dependent switch driven by this DNA binding protein. So we have two switches at this point I've described to you. One is this nucleation, which gives metastable silencing, which lasts about 10 days. We can actually, with mutants, separate just the nucleation from the spreading, and that you can show that that nucleation event confers a metastable silencing, which is then fixed into a long-term epigenetically silent state by now spreading of the polycomb over the locus. Okay. So one last little bit of mechanism. So that's about memory, okay? How you actually generate that polycomb memory is thinking about the cold. Where does the temperature come into this? And I'll get back to that because of the natural variation at the end. So the only cold step I've told you so far is this induction of this PhD protein, VIN3. But when we looked in a VIN3 mutant, completely defective in this protein, and this is what FLC does. It sort of starts to go down quite normally. You then just can't hold the silencing. So that suggested there were other cold steps for earlier on. As we went searching for those some years ago now, we found a whole series of antisense transcripts to FLC. Because hot air was the rage in the sort of homeobox silencing in mammals, this was christened cool air, which everyone remembers the name, so that's what we call them. It's actually a series of transcripts which initiate from basically the end of the sense stop site. There's a promoter, and these are alternatively spliced and alternatively polydenylated and form a series of these, which actually all have quite independent functions, but they are all induced by the cold. Now, I rushed through this. A lot of this has been published, but our latest data wanted to understand how are they working, because the effect of the antisense seemed to be independent of the actual polycomb silencing. And so in this paper, what we showed, and this was by using single molecule fish, where we label either the antisense in green with these probes, which are the green probes here, or red, which we're looking at the sense intronic nascent transcript, in a nucleus. So you can either see in the same spread, either the antisense is made or the sense is made. But you never see sense and antisense at the same locus. So as you induce the antisense, what happens then is you switch off the sense. And this shows it very nicely here. So we're looking very hard at the moment at what on earth in the cold induces antisense. But as you induce these antisense transcripts, you transcriptionally repress the sense transcription. So you've got a series of complex regulatory things determining quantitative level, which also involves the antisense. But just for this talk, the cold sensing involves then this big induction of the antisense transcripts, which is important to shut transcription down. And that shut down of transcription is important for nucleation. You can't have the polycomb nucleation if you have transcription, because for some modeling work that probably Ben has read, but I'm not going to have time to get into here, we see transcription as directly the antagonistic process to polycomb silencing, that there is mutual exclusivity with transcription and polycomb antagonistic to each other. That is actually involved with a physical clustering part in the nucleus. This is in this Rosa paper. That keeps it off, and then there's, as I said, a spreading of the polycomb as the plants come into the warm to maintain that off state in a very robust silencing through the cell divisions required for allow the plant to flower. And just as an aside, and what was interesting when we then looked, this is reset every generation FLC, because of course every generation of seedlings needs to have a vernalization requirement if it's going to work. And in fact, the first mutant we got out of the resetting encoded a demethylase, so that was satisfying. Okay, that was a sort of whistle-stop tour through the mechanism, because I want to now go back and look at the natural variation in this with this mechanistic understanding. So where we got to was that there are these FLC types which seem to have a phenotype, and they're distributed around the world. So I'm going to talk about two that we know so far. This one here, this haplotype, and this one here. So this one is the first one. It was collected from northern Sweden. There's quite a small number of these. But actually, when we looked, compared to, this is a normal profile I showed you in a rapid vernalizing type, what happened with this Lervik accession was that the FLC at the beginning looked just the same, actually during the cull looked just the same, but what happened with a 4-week vernalization, there was not any epigenetic silencing, the gene was reactivated. So there was nothing wrong with the sort of mechanism itself, because if you gave the plant 12 weeks, then it was perfectly epigenetically silenced. So as we've gone in, and I've shown the schematics now, so this is what sequence of events. Here's our nucleation in a rapid vernalizing type with 4 weeks cold. Either 10 or 30 days after you take them back out, you have a nucleation in the cold, and then the spreading and the stable maintenance of that silencing for 10 and 30 days post-transfer. If you look at this slow vernalizing type, this LERV1, for 10 days, it's absolutely the same. But if you leave it a bit longer, for 30 days, what happens is somehow this robust silencing is lost, and actually the gene then switches back on. But it takes a while for it to switch back on because this metastable silencing is intact. Now, if you give it long enough cold, then the silencing, the long-term silencing, is quite stable. So there's something about how long it's in the cold that's basically affecting the ability to transfer from a metastable to a long-term silencing state. And when we went in, and so here's our rapid vernalizing sort of standard Columbia type, and we look at all the SNPs that distinguish these two. And then we did lots of mix and matches, and the first experiments were shown in this 2012 paper, and we've just begun to finalize this now. It's these 4 SNPs here that are causative for this difference. And as we've gone in, I think this is the one that is the most important one. And I point this out because it's sitting in between those 2 RY elements, which is where that DNA binding protein interacts. So now we're trying to really understand, actually what this SNP is. Presumably it's affecting the dynamics of an association of one of the trans factors, which is then affecting the ability to switch from metastable to long-term silencing. Now, what's interesting is this activation or reactivation in this accession is relatively slow. But it is, again, reminiscent of a sort of on-off state, because if you look, you can see patches of cells where the gene has been switched back on next to patches which are still silent. So just like we have the sort of switching off mechanism we have when it comes back on, it's stochastic switching on. It's also relatively slow, and we do this sort of quantitative analysis, and Ria Ateneu-Kurinotu has been doing this with Martin Howard. They've been trying to sort of work out from the actual data how many switches are required. Now, I've shown you 2 so far switching. You've got the metastable switching and the long-term switching. That doesn't seem to be enough. So in fact, from the really detailed analysis of the reactivation dynamics, it looks like there's actually another phase of the memory phase where we lose this metastable memory altogether, and the plant then absolutely relies on just the spread state. So you're not getting any delivery of proteins. And this becomes important for the next phase, because now what we're trying to do is really model. Here we know these PhD proteins are binding. They stay for about 10 days, and then they dissipate, but they transfer to long-term memory. And this now can actually survive just with probably histone K27 feedbacks being sufficient. So we get into real sort of understanding of polycomb silencing. Okay, so that was one. That's where we are with that one. How about another haplotype here? This one, again, tends to be from northern Sweden. This one was interesting in that when we looked in Norwich, and this was done in a greenhouse over a winter, so this is the sort of classic winter profile in 2014-15 winter. And we had actually got this accession and a near isogenic line where the only difference between that and Columbia was the FLC allele. You can see that this is what the normal Columbia looks like, And this is now the nil, which looks very much like the accession. So it's very, very much later flowering because it's got very much higher starting FLC levels, and they take much longer to slow down. Now, when we look at the SNPs in that particular haplotype, in fact, the one that caught our eye was one that was very close to the processing of one of the Coulet transcripts, the splice site here. It was right next door to the dinucleotide that's adjacent to the splice site. And in fact, indeed, as we looked at this, we found that this changes the splice site. In fact, it's shifted 54 bases downstream, and an additional exon is brought in just because of that one single SNP. We can then recreate just that single SNP in this type, and you get the same phenotype. So that single SNP is enough to give you late flowering and a slower vernalization response. Now, we wanted to understand what that non-coding transcript was doing. And in fact, Carissa Sanbomatsu, who's someone who works at Los Alamos, had heard me give a talk and came up and said, well, this is really interesting what this non-coding RNA is doing. Would you like me to do the secondary structure? It's like, great, so this is the in vitro secondary structure of the different forms of that Coulet splice transcript. This is the one from the rapid vernalizing type, and this is the one shape mapped, so you attack in vitro with chemicals and then ask where you've managed to attack. This is the one from the single SNP change, and you can see the biggest change is in this helix here. Now, as we go in and look actually at a whole series through the Brassicaceae, you can see that this rather complex structure is actually conserved through many millions of years of evolution. And what's interesting is that this is really, a large part of this is intronic sequence. So there's very little sequence conservation. So we have a structural conservation with no sequence conservation, suggesting that there's some important function for the sequence. Now, so far what we know, for the structure, so far what we know is that non-coding transcript influences the capping levels of the sense transcript and these chromatin modifications. And we're still trying to understand how on earth an antisense non-coding transcript influences the sense transcription, because that is what this natural variation is changing. Now, what's interesting too, and I put this in from an evolutionary point of view, if you look in other brassicas have more than one FLC copy. In fact, this is Brassica rapa with 4. There are 4 rather different copies. You see the tree here. They're expressed differently, so A10 is certainly the most abundant one. And what's amazing to me is that has this same alternative splicing of the Coulet transcript as the FLC accession, but quite different sequence, so evolved independently. And that is also associated with higher expression and with this little short stunted helix 4. So we're now trying to work out what exactly this is binding to, because presumably the structure conserved means it's binding to something, and how that impacts expression. Now, the last little bit of the talk is really to go back and say, okay, we've got this natural variation, and we've sort of got the mechanism, and we're beginning to unpick how the natural variation is affecting that mechanism, and it's affecting it in very different places, and we've only managed to look at 2 so far. But what is the importance of these changes actually out there in the natural world? So we then embarked on field experiments, which is a hoot, frankly. You're used to doing lab work, and then you have to do field work. It's quite a different experience. And so I put a few of these pictures in here just to show the disasters that we had. They were basically northern Sweden, right up in the high coast, absolutely beautiful. This is where this Lurvik type was. So we wanted to actually go to sites that where we'd collected the accessions from, their natural homes. There was one in the south, a site called Ulstorp, which turns out to be Magnus's uncle's farm. Very useful to have contacts in Sweden. And then we had this site in Norwich, which was a sort of ransacked greenhouse where we could do some transgenic work as well. And we had all sorts of disasters, mudslides, it rained a lot. But I have to say, I'm enormously grateful. This was an ERC project that allowed us to have enough money to do this and work with someone in the University of Mid-Sweden who was up here, and then Torbjörn Sahl and Lund down here, so that we had master students who were prepared to go out. We sewed them all, and then every week they would go and harvest and send the material back to Norwich, so we could just receive them and then do all the FLC expression analysis. So we certainly had the easier bit. But we had some wonderful retreats, and they were very excited to be part of this, really trying to understand, how these FLC alleles were different in the different winters. Plus, we've done a series of now if you like, parameterization experiments with controlled environment rooms where we then try and mimic what's going on in the field and try and understand exactly how the plants are incorporating the temperature. So there's a vast amount of data. We're working our way through it. This is definitely sort of still ongoing. This is just for one accession. Okay, this is our standard, bog standard Frigida columbia accession, grown in Norwich. There were 2 sowings in Sweden because we weren't 100% sure how it was going to work, but that's actually quite nice because they do look vaguely similar, and they were sown 2 weeks apart, so it kind of gives you a bit of a nice internal control. And then southern Sweden. And so if you stare at them, you think, okay, well, Vin3, do you remember this is this cold-induced pH regulator required for the silencing? Well, that goes up in the field, so that sense differently, depending on the different temperature exposures. FLC comes down, so fortunately, at least in the field, it's doing sort of what it's supposed to be doing. But then when you stare at this, and these are log plots for FLC, see the decay is sort of fairly linear on a log plot in Sweden. But actually, if you look in Norwich, I'm sorry, this is a bit faint, this is the sort of the log linear plot to try and sort of put a number on this. But actually, when you look, it almost looked like there was a different phase early on here. And we realized this was really, Vin3 had not been induced. And this happened to be the rare occasional warm autumn in Norwich. I forget, it was which year, 2014, 2015. It just happened to be very, very warm. We were rather lucky. And because of that, we'd actually captured 2 phases. So this is the same data. This sort of squiggly is the real data. So we've actually got 1 rate of silencing and then a second rate of silencing. And we reckon that was linked with the Vin3 expression. And in fact, we had the Vin3 mutant out there. And indeed, the Vin3 mutant stays at this rate of silencing. But then when Vin3 is turned on, now it switches to a different rate of silencing. So now we could actually start to look at this. This has just come out. So we have different dynamics, and this is actually sort of this upstream transcriptional. Remember all of the stuff that you need to shut transcription down before you can polycomb silent. This is important in warm autumns, and then you have the polycomb dependent, the Vin3 dependent one later. Now we go in with, and I whiz through this fairly quickly, with now trying to recreate some of these different temperature profiles to try and unpick just how the temperature's inputting into this mechanism. And we do that by giving the plants either constant temperatures, but over a long, over a long period of sort of 6 or 8 weeks, of 14 fluctuating or 14 constant over a long period, or 8 degrees fluctuating or 8 degrees constant, to actually start to ask which bit of the temperature profile is important. It turns out for Vin3, 8 degrees fluctuating or constant, you get induction very similarly. But actually for the fluctuating, this one here, sorry, which way around have I got here? This is the constant, and this is the fluctuating. In fluctuating 14, you really can't accumulate your Vin3. And a similar sort of set of data with looking at FLC transcriptional shutdown, again with these different fluctuating and constant. What that says is that you can't shut down FLC unless you have fluctuating, because the 14 constant, you're not getting any shutdown. So we're starting to sort of unpick really interesting aspects of the temperature input here. And so to summarize that, what we've got, and this is the real temperature data, we've got different phases where this part of the temperature, and this is anything below about 10 degrees, is actually driving the Vin3 independent, and this is where the antisense comes in, transcriptional shutdown. And you need these low temperatures. So a constant temperature here isn't going to trigger this. You need temperatures below 0 to actually get this to be triggered. You then have below 15 triggering of the Vin3 induction, which leads to the epigenetic silencing. But for that first set of graphs I showed you, what you mustn't have are temperatures above 15, because anything above 15 turns over all the Vin3 you've accumulated. So the plant is actually monitoring the peaks and the troughs in slightly different ways. It monitors the coldest bit of the night to influence the silencing of the antisense, to transcriptionally silence, and it monitors that in the afternoons, peak afternoons, this temperature is low enough to actually think, yes, it's really now winter versus autumn. Am I not going above 15? Well, that was a surprise to me. I don't know. I just thought this was, because for a long time, if you look in the literature, everyone's just just thought what plants we're going to be monitoring was a day degree kind of something accumulating slowly with a certain temperature profile. But this really looks quite different. And of course, with our natural variation, we've got the LOVE1 is influenced in this aspect, the SNP that affects the long-term silencing. And this one here, this SV4, is influenced in this one here, in the low temperature perception. Now, this just shows lots of data to show that we've got lots of data. It's hard to actually begin to sort of unpick it all. This is now all the natural variants in all the different sites. The two obvious things that jump out is actually the rate of silencing, long-term silencing, is really quite different in the north and the south. The two north look pretty much the same, happily. But you also see lots of different variations. So now we're really in the middle of trying to unpick all of this to understand, we've really now got differences at different sites and each haplotype is responding differently. And of course, each year, the temperature profile is slightly different. So not one strategy is going to be beneficial for any particular year. So we've got a complicated story, but hopefully we're getting to the point now where we understand what the SNPs are doing to the mechanism, and we know which bit of the mechanism follows which temperature, so we can really get to that mechanistic understanding of how those SNPs are influencing FLC silencing in that location. Now, I just want to sort of pull out a bit and sort of say, okay, I started off saying, gosh, there are an awful lot of genes that regulate flowering, and the expectation, certainly Magnus's expectation, was that adaptation to really a very wide range would have involved changes in lots of different genes for all sorts of different selective pressure. But in fact, we haven't found that. What we've found is that there seems to have been really major nodes, and FLC is one of those. So why could that be? And it's also seemingly true in the broccolis cauliflowers, the Brassica oleracea that we've looked at so far. Now, that could be because of this slightly weird sort of circuitry where you've got sense and antisense at FLC. And what I've shown you is that, changes in various aspects of the antisense feed back and affect the sense transcription. And these loops tend to magnify any change that we see. So you've got a large area where you can accumulate changes which can functionally sort of affect the sense transcript. It's regulated both in cis and in trans by various factors. And of course, any change, because of this quantitativeness, has a directly effect on flowering and therefore very strongly affected in terms of reproductive success. So potentially this has become a node because of this really unusual sense-antisense circuitry and this epigenetic regulation. But the other thing which was, sort of like, well, somehow, and this is what I'm looking forward to hearing about at this meeting. So as I say, the expectation at the outset of this was really that we would see lots of genes that were being used in evolution, lots of small changes were going to be contributing to sort of a trait as important as flowering time. And so, the FLC didn't fit with that. But also, when we looked at this other pathway that we've looked at in the plants, and this is what actually sets FLC expression in the first place, and this was some really old data I just wanted to kind of highlight again. One of the first genes that we cloned was this upregulator of FLC, which effectively makes the plant a winter annual. And when we cloned this out, it's 2000, and then looked at the mutations in the rapid cyclers, I think we're now up to 25 independent loss of function mutations in Frigida that have caused the winter annual to rapid cycling evolutionary step. So again, this whole step of this life history strategy, which is a really fairly fundamental difference from a plant, whether you're winter annual or rapid cycling, has come through a recurrent loss of the same gene affecting FLC again. And when we look at the sort of flowering time generally versus in the different Arabidopsis and Brassicaceae species, Arabidopsis thaliana has sort of got a much broader climate envelope. So it's almost, it's like to speculate that you've got this variation in vernalization, which has taken it to a certain part of the climate envelope. And then the last step in evolution has now been perhaps to take you to rapid cycling, which has then opened up a whole nother set of different geographical range for you, climate envelope range. Anyway, I throw that in as terribly speculative just to start a bit of conversation. So let me just sum up. We started off really a long time ago now by trying to understand the seasonal timing and the molecular basis of it and how that might have changed through natural variation and adaptation. Unexpectedly, we've found that that's really centered on a few major loci. We've focused in on FLC and looked at the mechanism. We've got SNPs affecting epigenetic silencing or antisense transcripts, so those SNPs affect very different parts of that mechanism. We've got lots of inputs of temperature. Temperature, there isn't just one input. For a long time, people thought there was a temperature input. Everywhere we look in this pathway, the temperature has an influence. And it's rather different. Sometimes it's the high temperature, sometimes it's the low temperature, sometimes it's the average temperature. The plant is integrating a lot of different aspects to work out what is going on over a seasonal time. And then, again, we've got, as well as the sort of predominance of FLC, we've got rapid cycling winter to spring habit evolution through recurrent loss of one of the activators, the gene called frigida. And this just shows some of the people involved in the field experiments, actually, diversity, great, and this is what the Royal Society encourages us all to do, to sort of anti-Brexit logo. But it's sort of, my team is made up of a lot of different people, and long may that last. So we've got people, these are the Swedish people who are based in Sweden. Svante Holm really spearheaded a lot of the field stuff for us. I'm very grateful for him for doing that. The people this is Rhea, who's done a lot of the unpublished modelling, and Joe, who masterminded the field work. And I just want to point out Martin Howard, who's the computational biologist too. We started about 7 years ago, and it's actually a fantastic collaboration because exactly we get into sort of quantitativeness, and he looks at graphs. He says, Caroline, I think you should look at this, and then I go away and do the experiments. And so it's been a fantastic collaboration. And our funders. And thank you for your attention.
Okay, questions?
It's great to hear the full picture. It's impressive to see how much you've managed to figure out at this locus. And I was wondering what, in your eyes, is the strongest evidence that the variation you see at the FLC locus is really adaptive.
Yeah.
Because when we grow all these lines in our field site in Cologne, they tend to flower within a week of time, the ones that are FLC that are require vernalization, and those that don't sort of roughly flower within a few days. And so, I'm interested to hear that.
I think this is what's coming out of this really unusual warm autumn. So most winters, probably most things will flower really roughly the same. But for example, that 2014 winter was in Norwich was very warm. That changed the profile. Last year, there was a very, very warm Arctic winter. In fact, some of the native plants flowered in northern Sweden before the snow. You remember the seed bank flooded. So I think what happens is that those winters now confer huge selection for certain strategies. And you've got to have very high FLC when you're, for some reason, your autumn's slightly warmer. You absolutely want to keep flowering off until the snow has come. So I think for most winters, perhaps, you won't see an awful lot of fitness differences. And then that weird winter will lob in, or like this spring, very late. That's going to affect everything again. And so some strategies will be better than others. And so I think, you'd have to do a very long-term analysis to actually be able to unpick some of that. But I think the fact that we can now go in and know sort of exactly the SNPs, we've got the nils, we can simplify it down to actually try and get an answer to say how adaptively important these are. Plus, actually, a colleague of mine at John Innes, he finds that actually when the plants flower affects their dormancy levels, whether they go to primary or secondary dormancy. So, we're just looking at flowering time, but of course FLC is more than just flowering time. It's actually the sort of coordinator of many developmental transitions, and then you'd have to put all of that together. So quite a hard question to answer, actually.
Hi, I've got a question about maybe a bit detailed, but about the Lervik one comparison contrast with Columbia. You mentioned that there's actually this, in your schematic, you talk about actually having a spread H3K27 trimethylation signal. I guess my question is, and then it seems to be lost a little bit.
Yeah.
And I wonder about the whether there's actually a role here in terms of the experimental assay itself, because you also mentioned that FT seems to be averaging the signal across different cell lineages, right? So in the sense of whether the chip assay for looking at the trimethylation mark might be actually less lineage sensitive, and so I'm wondering whether the detection method actually seemed to, I guess in my mind it's hard for having a spread repression signal be erased for the short, for FLC to come back. So just maybe can you
Yeah, I
comment on
didn't
that
put it
contrast
in.
between
I can show you the data. Actually, you basically look and you see accumulating levels over the body as you give more and more cold. And then for 10 days and 20 days, that's quite high. And then suddenly at 30, over the body, it's gone away. Now, we also look at the nucleation region, and that also decays. And even in the rapid vernalizing types, the nucleation of the proteins at the nucleation site only lasts a certain amount of time. And by 20 days after transfer, even those have gone away as well. So in fact, yes, you have these sort of mixed populations, but actually the chip assay works quite well because you're just grinding everything up and averaging everything out. And you can then look at the quantitative changes on that basis. Plus then you can actually look at I showed you the reactivation, which is in patches.
You talked a lot about mechanism in the sense of regulatory molecules, but there's of course a deeper layer of mechanism which is sensing the physical temperature and translating that into molecular output. Do you know whether there's also genetic variation in that, or is that too hardwired?
Yes, now that would be really nice. I mean, I would love to actually know one of the primary cold sensing steps, because actually for a long time people thought there was one or perhaps two major kind of cold inputs. We're doing a lot of work at the moment. I think we're up to 7 different inputs, and they all have slightly different patterns of behavior. So then we'd have to define each of those to then look very specifically for natural variation in that, depending on what that is. So we haven't unpicked it enough yet to actually know that. But certainly there's no obvious natural variation that we know of from what I've said so far that you'd say, yes, that's just I feel hotter or I feel colder kind of variant that we know of so far.
And the mechanism is rates of enzymatic...
Well, the mechanism for, let's say, the nucleation, so that's temperature sensitive, you might imagine would be a kind of on-off rate on the kind of chromatin itself. For Vin3 induction, we've actually got a whole series, which is that PhD protein, trying to understand, we've got mutants which now influence that and go in. And that's taken us into the world of, there's a transcription factor which has got a transmembrane domain which is required for the cold upregulation of Vin3. So then we have to get into, have we got some transmembrane domain release temperature dependent mechanism? So, we keep going back, but yet we haven't got to something I can say, that's the actual temperature step yet. It's so, and then there's RNA, those different RNA molecules. It would be lovely to sort of, some of the rock silencing with Asifa Akhtar's work sort of suggests that maybe, you're going to have some kind of temperature dependent RNA structure. But there's nothing I can put my finger on yet and tell you that's the temperature step. But I think it'll be working through lots of different, temperature affects biochemistry in every which way, and that's what we've got, an integrated system.
I was wondering if you mentioned you didn't see any latitudinal correlation in this natural variation, but did you look at the stability of winters in terms of more continental winters to more oceanic winters? Do you see any correlation there?
We've not done a lot of that, because you kind of need to sort of have a lot of I think you'd need a lot of the data, the expression data as well, to really make some sense of it. So we are trying to do predictions for future winters, because kind of climate change scenario type things. But we've not, there's a lot of data. You could sort of do lots of kind of data moving around, but that's not my kind of, so not a lot, actually.
So these recurrent deletions in Frigida, which
Yeah.
are a great example of deletions in a gene that's conserved in different species. So I didn't realize. So the other Arabidopsis species don't lose this. In the same,
So,
I mean, it's not polymorphic.
So in Thaliana, there are 2 clear deletions and lots of stop codons. So that I whizzed through rather fast, but they're protein stops. So far in the other Arabidopsis types, there doesn't seem to be Frigida nulls that actually account for flying time variation. But it's been interesting that the Arabidopsis thaliana gene has translocated onto a different chromosome compared to all of the other Brassicaceae. So it's now near the nuclear organizing region on chromosome 4 with a different promoter. So we published that a couple of years ago. So I think that's sort of affecting the likelihood of it being a target for sort of this natural variation. But what we have found, when so let's say in Brassica napus, we can knock out Frigida and apparently have no phenotypic effect, but there are multiple copies. But then we've realized that as we knock them out, the other ones, the expression goes up. So you actually have to knock all of them out, and now you start to see a phenotype. So as with all these things, when you sort of knock one out and don't see a phenotype, you think, oh, well, it's not important. But actually, that was the wrong answer.
I have a very related question. So it's very impressive that you found all these sort of mechanistically convergent different mutations that cause these phenotypes. I would just wondered if you have a sense of how old this variation is.
We haven't really done that. That's the kind of thing I keep saying to Magnus. Surely we should be doing stuff like that, Magnus. So I don't really know. There's a lot of data. I am not sort of someone who does a lot of this. So, if it looks like an interesting question, there's lots of data to ask that. But they're single SNPs. I mean, I don't know quite how you would do it. We've got the haplotype data I showed you. It's a single SNP which distinguishes them. So we haven't done that. We've got data probably could come up with some sort of answer, but we don't know. None of the, I guess we could go to Lyrata and all of these other relatives. They're not in any of those. So it's not that old. The Brassica, the SNP I showed you that affect the intron splicing, same functional thing, but no, it's not the same SNP.
You mentioned that the network had been rewired in different species. Could it be that in other species, it's other genes that have evolved such complex regulatory mechanisms?
Yeah, well, I think, wheat's an interesting one because our institutes always wants us to work on wheat. And when people first analysed the winter-spring difference in wheat, it wasn't a MADS gene. They went, Caroline's wrong. It's completely different in wheat. And there, I think it's VERN1. It's an activator, actually. It's a MADS activator rather than a repressor. But actually, after many, many years, what's happened is that, there's another one called CCA, CCT type gene that's important in wheat. It's the same sort of mechanisms, but it has used slightly different components that are all present in Arabidopsis. It's just different ones have been chosen, if you like, as the evolutionary node. I think that's what's happened.